Marcin Cislek. Many of you all probably already know him. He's been collaborating with several folks in the department, and he's here at Michigan as a research investigator on the faculty with the Michigan Center for Translational Pathology, where he works with the Rule and the team over there. And he's becoming a leader very quickly in a very important area that bridges therapeutic discovery all the way out to treatment, and depends on bioinformatics analysis, including novel methods applied to immunological components to help us understand the very important area of cancer immunotherapeutics and immunotherapy, both at the genotype level, phenotype, and even into outcomes. So it's really an impressive array of capabilities that Marcin brings forward to us. He is I think in your heart of hearts, you're not only a computational biologist, but a biophysicist, which, since I was trained as a biophysicist, that always makes me happy. He likes crystallography, things like that. He also is a biotechnologist, got a master's degree in biotechnology in Krakow in his native Poland, and then got his PhD in structural biology, biophysics, computational biology at the University of Virginia, Charlottesville, one of the most beautiful campuses on the planet. He's been with us since 2013. He's already a PI on two important venues in the prostate cancer area, Young Investigator Award in prostate cancer with the Prostate Cancer Foundation, and he's also has a 3-year award out to 2020 with the DoD PCRP program, looking at regulatory networks of immune evasion in metastatic prostate cancer. You know, I could go on, but I think it would be better to let him go on because we're really in for a treat. The research is cutting edge. He already has, you know, nearly 20 first author papers and another bunch of collaborative papers. And, you know, he's really making a mark for himself at this early stage in your career. He is a faculty recruit here in DCMB. We're proud to say that. And we welcome you to DCMB and to our seminar series. And we're excited to hear about your exciting work. Thanks for coming.
Thank you very much. So, thank you very much for having me. I had a very nice time. Thank you for the students for meeting with me. Thank you for the faculty who are taking their time to meet with me over the next 2 days. So what I'll talk to you today is a little bit different from what Brian introduced me with. What? It worked. No, the pointer doesn't work. Okay, now. So I'm interviewing simultaneously at the pathology department and at the bioinformatics department. And when I was presenting a talk to the pathology department, I thought it was very important to root my bioinformatics research clinically to the applications of and the needs of oncologists, of pathologists, of medical school. Since I'm presenting here to a bioinformatics department, I decided to structure my talk a little bit differently. I will talk more about the algorithmic and the bioinformatic strengths that I come with and how they can be applied to study cancer and also how they can be translated. So I'll talk to you about the position effect as an organizing principle for studying somatic aberrations in cancer. And really the idea for this research and for this topic came about 3 years ago when I was like thinking, I was at that point studying structural rearrangements in cancer using RNA-seq data. And I had this idea and I was thinking, so when a structural variant occurs, what does happen to the chromatin that was at the point of the breakpoint? Does it change? Doesn't it change? Then I asked a senior member of our lab, Dan Robinson, whether he knew anything about this, since he's our go-to person for this type of knowledge. And he says, I don't know, but you should read about the position effect. That's how I learned about the position effect, and that's how I also frame our future discoveries in that area. So to motivate my research, I would like to show you how a cancer looks like in terms of its genomic organization. So a primary prostate cancer looks typically like that. The black line represents the 2-copy state of the genome, and as you can see, the genome is largely diploid. There are a number of small copy number aberrations. Most of them are very recurrent, like, for example, the gain of the 8q arm, the q arm of chromosome 8, or the loss of the 8p arm on the 8th chromosome. For example, the loss on 16q is also very prominent, but overall, there are very few large-scale changes, hundreds of breakpoints, sometimes only tens. On the other hand, a metastatic cancer looks like that. It's just haywire. Everything is rearranged. The cancer is baseline triploid. We have LOH, single copy, up to 5-copy whole chromosome arms, lots of structural variants, a mess. And what I find quite remarkable is that we don't really understand what the functional consequences are of this and how they are selected and how these rearrangements really contributes to the mission of a cancer to survive and take over. So obviously, all those copy number changes are also reflected in structural variants. We can use various techniques to discover structural variants. One of them is gene fusions. And as you can see, prostate cancer is among the most rearranged cancers based on the number of fusions per sample across all the other metastatic tumors, which is surprising because primary prostate cancer is among the least rearranged cancers. So just to introduce you to the position effect, and here I'm really just introducing the concept. It was discovered in 1930. It was discovered in Drosophila, where it was noted that ionizing radiation causes inversion on the Drosophila chromosomes, which can rearrange a gene. Here the gene is called white, and when the white gene gets inverted into a different locus, it stops making the pigment, so we have a very patterned coloring of the Drosophila eye. In molecular terms, the white gene gets inserted into heterochromatin, and then as a result of chromatin domain spreading, the gene gets silenced. But what I would like to emphasize is that this process is quite random. In some certain cells, the gene will be silenced. In certain cells, it won't. And sometimes a cell that got the gene silent will grow, meaning the silencing happened early. So we have those patterns of big blobs where the gene did or didn't get silenced, which points to the inheritance, the epigenetic inheritance of those changes. The position effect occurs also in animals. So here's an example of the steel locus in mouse, and a normal mouse looks like that. This is a mouse with an inversion. This is a mouse with an inversion, which puts the gene even farther from the enhancer the gene is regulated by. And this is a mouse which has a mutation in the gene itself. So as you can see, the position effect can have sometimes effect just as a knockout, and sometimes it's an intermediate effect between the wild type and the knockout. Of course, there are many genetic diseases which are associated with position effects, and they are often discussed as such. The list is quite long. I would like to focus here on two of them, the Hox-D locus rearrangements, and also the most frequent translocation that doesn't involve a whole chromosome arm in human. And this translocation is phenotypically silent, but creates a huge risk for the Emanuel syndrome due to missegregation of the rearranged translocated chromosomes. So just to show you the Hox-D locus, it's probably one of the greatest discoveries I at least have read about. So there are diseases associated with the rearrangement in the Hox locus. So here are 3 patients. All of them have a translocation or an inversion that breaks the Hox-D locus in half. And the phenotype of all of the patients, although slightly variable, looks somewhat like this. The arm forms, but the fingers of the patients do not form. If we look much later about the molecular biology that was done to study the Hox-D locus, and here really beautiful work from Dubel's lab, where they showed that the Hox-D locus is regulated by 2 different sets of enhancers. These enhancers regulate the locus in the forearm or the arm, and these enhancers regulate the expression of the HOXD genes in the digits. And obviously, if a translocation separates HOXD from its enhancers, then the digits will not form. There's also an underlying spreading of silencing marks on the chromatin that really causes this enhancer association switch between the HOXD locus. The other example I wanted to give you is the translocation between chromosome 11 and chromosome 12. And here, the translocation is phenotypically silent, but a very nice study, kind of ignored by the scientific community, showed that what they did, they created cell lines out of patients who had the translocation, and they looked for gene expression difference between the rearranged and non-rearranged cell lines. And what they found is that the majority of genes that are differentially expressed between those two sets of cell lines or two sets of patients are actually on the chromosome arm of the rearranged derivative chromosome 22. To me, this is absolutely remarkable, which means that this translocation, which puts one piece of chromosome onto another chromosome, has such wide-ranging consequences. The position effect actually extends way, way, way from the actual breakpoints, actually dysregulating the whole chromosome. So I would like to discuss position effects in the context of metastatic prostate cancer. And just to give you a very broad overview, metastatic cancer has many recurrent copy number changes. What I show here are either recurrent copy gains or recurrent copy losses. And as you can see, there are a number of genes which are associated with recurrent gains and recurrent losses. But also metastatic prostate cancer is associated with many different mutations. Among them, the most frequent ones are mutations in FOXA1, but also mutations in CDK12. And I'll discuss how position effects are related to mutations in FOXA1 and also mutations in CDK12. So just to show you mutations, CDK12 is recurrently mutated at about 7%. All of the mutations are inactivating, so they inactivate both alleles of CDK12. As a result, the tumor becomes CDK12 null. And the frequency of those mutations is much higher in metastatic castration-resistant prostate cancer than in primary prostate cancer. But there is one intriguing finding. The CDK12 gene is absolutely essential. So the loss of CDK12 is incompatible with life. Here, and this has been reproduced by many labs, including ours, but here's a published CRISPR knockout screens, which shows that CDK12 is among the most essential genes in the human genome. So of course, there's a question, how is it possible that the tumor loses both copies of CDK12, but still manages to survive? The CDK12 loss results in a very dramatic copy number phenotype. So here you can see the copy number plot for a select CDK12 case. And as you can see, it's baseline diploid with very few copy losses, here highlighted in blue, but a large number of focal tandem duplications like this, here highlighted by those different spikes. We can look at this at the ploidy plot, and you can see that compared to other types of genomic aberrations, such as homologous recombination deficiency or mismatch repair deficiency, CDK12 mutant tumors have a ploidy between 2 and 3, but really this means that they are baseline diploid plus 20% of the genome is in those focal gains. And the number of those focal gains, at least estimated from whole exome sequencing, is about 200 to 300. So there's quite a lot of those rearrangements. Right now, we don't know whether they form all at once or whether they are progressively acquired by the tumor.
So those little purple dots above, those are.
Where's the purple?
These?
Yeah.
Yeah. Follows up.
Yeah. So these are baseline triploid cases. So they have a baseline triploid genome, and on top of this baseline triploid genome, they have additional copy number changes. So then we looked at this from the perspective of whole genome sequences. Here I'm showing you chromosome 8 in this top bar. You can see here the different copy number states. So this is 1 copy, this is 2 copies. And as you can see in this particular case, we have this characteristic imbalance in prostate cancer between actually I mislabeled this, 8p loss and 8q gain. So the 8q is baseline triploid, the 8p was baseline diploid, but lost also 1 copy. And you can see those gains. The dotted lines represent structural variants, and you can see the structural variants are at the bookends of the copy gains, which allows us to determine that there are indeed tandem duplications. If we look at the size distributions of those focal gains, here CDK12, all our whole genome sequences for CDK12 cases are pooled, and as you can see, the size distribution is bimodal. It has a peak at around 300 kb and another peak at slightly over 2 megabases. If we contrast that to aberrations or tandem duplications seen in other types of tumors, such as homologous recombination deficiency, it doesn't really line up. However, the size distribution is quite similar to the size of replication domains, at least the one which are called in prostate cell lines. Here, the second peak is not very prominent, but the first peak, which is the most frequent size of a replication domain, matches up almost exactly with the CDK12. We don't know yet much about the mechanism that underlies the structural rearrangements, how it is stitched together, but some analysis of the breakpoint suggests that there are many inserted bases at the breakpoint. So there's some non-templated, possibly non-templated insertion or templated from a remote region. And this suggests a particular stitching together mechanism, but this remains to be, the details of that remain to be determined. So to just put you this in perspective, we have right now a number of classes of genes that determine genomic instability. There is homologous recombination deficiency, which is caused by mutations in BRCA, POLB2, or ATM, which leads to a phenotype which we refer to as BRCAness. It has a very strong number of characteristics. There are large copy number changes, aneuploidies, large-scale transitions, which are kind of big breaks in the chromosomes, and also a mutational signature, one of the Sanger mutational signatures called 3. There is mismatch repair deficiency. It's also caused by homozygous deletions of a number of genes, and this causes microsatellite instability and overall hypermutation with a specific signature. On the other hand, we believe that there's some form of DNA replication deficiency associated with CDK12 loss, which leads to this very characteristic phenotype of focal tandem duplication. So one thing that we thought of is that, well, since those tandem duplications exist, they should also generate fusions. Fortunately, we had a pipeline in place that was able to detect all of those different types of rearrangements. And here I would like to highlight that we are quite sensitively are able to detect passenger fusions, which might not be consequential, but they do generate neoantigens, which might be actionable in the clinical setting. So if we look at the number of fusions in those cases, we can indeed see that the CDK12 mutant cases have a high number of fusions, that the fusions are typically characterized by, detected by our algorithms as duplications within one cytoband, confirming the size distribution from the whole exome sequencing and from the whole genome sequencing. And again, this can be contrasted to the typical fusions observed in homologous recombination deficient tumors, which are typically translocation between different chromosomes. And there are relatively fewer duplications and definitely fewer than CDK12. I highlight this because there's a controversy in the field whether actually CDK12 loss has its functional impact through a deficiency in homologous recombination. Our results strongly suggest that homologous recombination deficiency and CDK12 loss are two very distinct phenomena. So as I mentioned, we can detect the fusions, and the fusions have many passenger events, and the passenger events might generate neoantigens. And here, with Pankaj's help from the lab, we built a neoantigen prediction pipeline, which starts with patient sequencing data, the RNA, the fusions, and goes through the pipeline of neoantigen prediction. to figure out what might be some of the functional consequences of those rearrangements. I implemented an algorithm to allow this particular prediction, meaning how to go from the rearrangements, possibly assembling across multiple breakpoints and translating a hypothetical chimeric protein so that we can generate the peptides and then rank them based on their predicted affinity to binding to the MHC class 1 locus. So if we look at the data, and here this data was generated by Pankaj, we can see that that CDK12 loss results in an increased neoantigen burden. It's increased relative to all the other classes of genomic instability. It doesn't quite match up with mismatch repair deficiency, but mismatch repair deficiency have hypermutations, MSI, and they overall generate a large number of neoantigens. And in CDK12 deficient cases, almost all of the neoantigens arise from fusions. So this is important because right now fusions are ignored largely as a source of neoantigens. Our results suggest that they should be strongly considered and incorporated whenever neoantigen vaccines, anti-cancer vaccines are being made. So the next question was, obviously, the fact that CDK12 mutant tumors have neoantigens might help in the somehow influencing their treatment, but it doesn't tell us much about the rearrangements. So the next thing we asked is whether those rearrangements are recurrent and random. So the first question is whether they are random. It looks like we didn't find any strong association with any type of genomic signal. They look to be randomly distributed. The only thing that was slightly significant is that they tend to be enriched in genic versus intergenic regions. But it's a rather small statistical association. Whether they are recurrent, well, we asked this question by developing a statistical model. And here I used a permutation-based model where I kind of tried to simulate what those rearrangements look like, how they end up being positioned on the genome. This allowed me to define a significance cutoff, and then at an FDR of 3.5%, I was able to call here the black lines, call the regions that seem to have a higher statistically significant enrichment for the CDK12 associated tandem duplications. I made this model in two different ways: a broad model, which considered all tandem duplications below 10 megabases, and a narrow model, which focused only on those below 2.5 megabases. Obviously, they detect slightly different events, but as you can see, the overlap is quite large. So when we look in detail on the most recurrent CDK12 loss-associated gains, we found a striking thing. Almost all of the gains were associated with, almost all of the cases had focal tandem duplications in genes associated with the cell cycle. So we found CDK18, MCM7, cyclin D1, and AR as a positive control of a locus that is under positive selection in prostate cancer. So if we map those genes onto the cell cycle, you can see that all of those genes kind of help in cell cycle progression. And there is some evidence in the literature that all of them have both a role in DNA damage and a role in cell cycle, and some of them have a role in prostate cancer progression. So this data, based on our mining of the whole exome sequencing data, suggests that we might have found the missing piece that the cancer cells do to be able to grow under the condition of CDK12 knockout, because normal cells without those amplifications do not. So the next, when I saw this result, this gave me an idea that, well, maybe this is something that could be more generalizable. Maybe there are copy number changes that are conditional on the mutations in other types of genes. So I got the help from Yaja, who's a master's student at this department, and we looked at the first very obvious questions. Are there any copy number changes that are differential between ETS positive and ETS negative, or here wild type, prostate cancer? What Yaja found is that tumors, tumors which are ETS positive seem to have many more copy gains across the tumor. Here I highlighted the most striking places where ETS mutant prostate cancer tumors have more gains than ETS negative prostate tumors. But also we found a very specific region which is deleted in 80% of ETS negative prostate cancer. This is important. So although this deletion was known before, because obviously it affects 30% or more even of all prostate cancer cases, the fact that it's so strongly associated with ETS status was not known. This suggests some sort of genetic interaction between, because of this mutual exclusivity, between the loss of 6p and ETS fusions. So we decided to look at this a little bit deeper because there was an ongoing discussion in the literature whether actually ETS mutations cause genomic instability. So here we categorized all the tumors into those that are p53 wild type and ETS wild type. This is our wild type group. p53 wild type and ETS mutant. These are the cases which have ETS mutations but are wild type for TP53. The cases that are TP53 mutant and ETS wild type, and obviously the ones that are mutant for both of the genes. As you can see, there's a gradual progression in the number of copy number changes. Here, just to show you all the copy number changes, everything that is wild type or 2-copy diploid is represented in gray, and all the changes are either blue or red. And as you can see, while you're progressing and gaining additional mutations, the number of changes increases. The most dramatic changes associated with p53 mutations. So this really clearly shows that p53 really guards the integrity of the genome, that only when p53 is lost, and to some extent ETS is lost, ETS is gained, the genome becomes unstable. The association is particularly interpretable in the case of p53 because p53 is inactivated through mutations, rarely through copy number changes. So this orients the causality axis. We know that p53 mutations cause an increase in the number of copy number changes, directly or indirectly caused. For the ETS mutation, the association is still not fully clear. So of course, we wanted to also know what might be the effects of those copy number changes in the context of CDK12. I first went back to a study that I've published previously as part of a large group. And there we looked at copy number changes associated with a very rare kind of kidney cancer, mixed spindle cell carcinoma. As you can see, this cancer is characterized by a number of chromosomal deletions. The deletions typically involve whole chromosomes or at least chromosome arms, and they are very highly recurrent. So for example, one copy of chromosome 1 is lost in 100% of the cases. One copy of chromosome 13 is also lost in 100% of the cases. But these tumors provide a good model to study the transcriptional effect of copy number changes. What you can see here is that all of the genes on all of the chromosomes that are lost typically lose 50% of their expression level. This means that if a whole chromosome is lost, really this is a dosage-dependent effect. If you have one chromosome rather than two, you have essentially half of the expression. However, if we look at smaller changes in the genome, like focal CNVs at the MYC locus, regardless of how many copies are gained at this particular locus, the expression of MYC is quite variable, and you can see here that quite perplexing, there isn't even a trend, which means that to interpret how the copy number changes affect gene expression will be a little bit more complex. So in the case of CDK12, I decided to look at this in a more aggregated way. So I looked at all of the loci that are either diploid or have an additional copy or multiple additional copies due to the CDK12 associated FTDs. And as you can see, the more copies you gain, the more genes are differentially expressed. So here, as baseline, out of the 20,000 genes, about 2,000 genes are differentially expressed in this particular analysis. And as you gain additional copies, you increase your likelihood of being differentially expressed. And by 10 copies or more, you have 50% chance of being differentially expressed. A similar analysis holds up if we look at whether the focal tandem duplications are able to induce expression outliers. And here you can see that at baseline, the chance of being an outlier is very low, well, by the definition of outlier, but each additional copy of a CDK12 FTD increases the likelihood that a given gene will be an expression outlier. And sometimes those expression outliers are actually the secondary oncogenic driver of CDK12 mutant prostate cancer. Here, for example, one of the FTDs generated a KIAA BRAF fusion. This fusion is not typically observed in prostate cancer, but on the other hand is very frequent in a very, in a rare type of glioma, pilocytic astrocytoma. So we also generated other types of, or detected other types of FTD-associated secondary drivers. Here, another BRAF fusion and also a secondary ETV1 fusion. When I looked at this a little bit more broadly, one could see two different kind of ways how the focal tandem duplications can affect the expression of genes. If we look at the MAPK signaling pathway, which typically contributes, the genes in this pathway are typically oncogenic in most of the tumors, although they are not very strongly oncogenic. You can see kind of a dosage-dependent effect, where for all of the genes, there is some trend of higher expression associated with multiple copies gained. On the other hand, for the receptor tyrosine kinases, this trend didn't hold up, and here there were outliers, and these outliers might be actually the drivers of this cancer, but overall the outliers occurred rarely and were not associated strongly with additional copies. So they were not stronger as additional copies of the locus were gained. The perhaps most striking thing and most confusing thing about CDK12 is that although all those focal tandem duplication associated expression changes are rare and kind of random from tumor to tumor, the cancer has a striking transcriptional phenotype. So there's a number of genes that are differentially expressed in CDK12 mutant tumors compared to other types of tumors. Here you can see that overall CDK12 mutants have more differential genes than any other type of genetically determined prostate cancer. And here you can also see that the number of differential genes broken up by up and down genes is higher in CDK12 mutant tumors. But I would like to emphasize that the differential expression of those genes is not due to focal tandem duplications. It is because of something different. Those genes are not more frequently in the FTDs than the genes that are not differentially expressed. So with that, I would like to wrap up the CDK12 part, and I'll go on to the FOXA1 to provide a little bit more local view on the role of position effects in prostate cancer. So first of all, FOXA1 is among the most recurrently mutated genes in prostate cancer, and it's also a gene that is very misinterpreted. So most people attribute that FOXA1 is a tumor suppressor, and they figure this out because there is an enrichment of frameshift truncating mutations within FOXA1. However, if one looks more clearly at that, you can see that the truncating or frameshift mutations are clustered in a very specific position of the fork head, which is the DNA binding domain of FOXA1. We also found that actually the distribution of those mutations changes strongly depending if we divide the protein into two parts, the fork head domain proper and the C-terminal part, and the C-terminal part is very strongly enriched for frameshift mutations, whereas the mutations directly in the fork head domain are not anymore so enriched. And also one interesting aspect of FOXA1 biology is that the mutations occur only in hormone-driven tumors such as prostate cancer, bladder cancer, and the 3 types of breast cancer. So Abhijit Perolia from our lab generated perhaps the most striking ChIP-seq data set I've ever seen, where he showed that the truncating mutations of FOXA1 are not loss of function like everybody thought, but they are actually gain of function. But not only that, he also showed that they are dominant, meaning expressing the truncated FOXA1 protein gets rid of the wild type protein from the chromatin. He has many more of such striking results, and I'll not go into the detail, but I would like to just say that we have pretty good evidence that FOXA1 is actually an oncogene, and that the frameshift mutations, but also the other types of mutations, are gain of function. But we didn't, the association between FOXA1 and position effects or structural rearrangements didn't come that directly, though. Quite some time ago, I became interested in seeing how sensitive structural variant detection can be from RNA-seq data, given that at least for genes related to oncology, we achieved quite high sensitivity and specificity. So if we plot the number of RNA fusions relative to the number of DNA breakpoints detected either from whole genome sequencing or from whole exome sequencing, this particular plot is from whole exome sequencing, you can see that there is a linear trend. Obviously, the more DNA breakpoints there are, the more fusions we detect, but that actually the slope is pretty good. For each 100 breakpoints, we are able to detect approximately 25 fusions, which means that the sensitivity of RNA-seq is not that bad. And on the other hand, we have many more RNA-seq data sets available in the public domain and also internally to look for recurrences. So here's one plot that shows such a recurrence. Plotted as one chromosome, you can see all the rearrangements within a cohort. They are different. Sometimes they are duplications, sometimes they are translocations, sometimes they are inversions. But here you can see that there is a place on the chromosome which seems to have many more rearrangements than other. So this gave me the idea that maybe it would be possible to identify recurrently rearranged loci based on the aggregation of the thousands of RNA-seq experiments that we already have, and that this could be done immediately rather than waiting for next technology to allow to do it properly. So this is what we found. When applied to metastatic castration-resistant prostate cancer, we found quite a number of recurrently rearranged loci. And as you can see, the signal-to-noise ratio here is pretty high. So here's a positive control. You can see the fusion, the tempers to ERK fusion, highly recurrent. Also, we can see fusions related to AR amplifications, also expected in metastatic prostate cancer. Rearrangements of the MYC locus reported previously, but here what we found is that we see quite a number of rearrangements affecting the FOXA1 locus. This is if we zoom in onto that locus. Here's chromosome 14. This is the locus where FOXA1 lives. And you can see that all of the breakpoints detected from RNA-seq cluster around the FOXA1 gene. So those breakpoints have been previously described, at least a very small subset of them, namely MIPL ETV1 fusions. But I would like to emphasize that the breakpoints are much, much more abundant, that the ETV1 fusions account for a couple % of the total number of rearrangements. So this is a very prevalent thing. So if we look at the numbers, tempers to ERK, as expected, is rearranged in 50% of the metastatic castrate-resistant prostate cancers. MYC is rearranged in 40% of the cases, and this is followed by about 30% of rearrangements, which is a very high percentage for any type of genetic event. If we look whether these rearrangements associate with progression, you can see that the MYC rearrangements, these are very lowly rearranged in normal samples, then highly rearranged in primary tumors, and then there's a very small increase in metastatic tumors, whereas for FOXA1, the number of rearrangements drastically increases between primary and metastatic cancer, suggesting that it might be related to either disease progression or treatment. So to confirm this, since this was a discovery based on RNA-seq, and there are ways RNA-seq can be wrong due to, for example, transsplicing, we decided to confirm this at the level of the DNA, and here is Here is one approach using whole exome sequencing. What I try to do is to detect recurrently rearranged loci based on the density of breakpoints detected from whole exome sequencing. Here's how it would look like. We have a locus, we have a number of breakpoints. So in this particular locus, there would be 4 breakpoints, at least in the copy number plot. To show that this approach works, we can confirm the high breakpoint density in temporous to ERK positive, in temporous to ERK locus, high breakpoint density in the PTEN locus. This is explained because PTEN is among the most frequently deleted genes in prostate cancer, and also a high breakpoint density in the AR locus. Obviously, these breakpoints are associated with the AR amplifications. So seeing that the approach works, we could apply it then on chromosome 14 and try to confirm the finding from our RNA-seq screen. And as you can see, there is a very high breakpoint density of around the FOXA1 locus. And also you can see here on this kind of copy number plot that here's the position of FOXA1 and most of the breakpoints as detected in RNA-seq data cluster around that locus. We also confirmed this in whole genome sequencing. We sequenced 9 cases. Here is the FOXA1 locus. In red are tandem duplications. In green are translocations. And as you can see, there's a high density of breakpoints close to the FOXA1 locus. So that makes it obviously interesting. So why might this locus be so frequently rearranged? So first of all, the most striking thing about the FOXA1 locus is that it's one of the most conserved syntenic loci in the human genome. So this is how it looks like. The synteny block extends from PAX1 to FOXA1. In human, the locus was duplicated. So we have PAX9 and FOXA1 and PAX1 and FOXA2. This goes back way back to sea urchins, 500 million years of those genes occurring in the genome in this relative orientation. The other striking thing is if we zoom into this locus, just into the vicinity of FOXA1, we have FOXA1, TTC6, its right flanking gene, and MIPL1, its left flanking gene. We see a number of highly conserved regulatory elements. They really show conservation really down way beyond vertebrates. If we look at this from a more topological perspective, this locus, quite similar to the Hox-D locus, is divided in 2 topological domains. So here the synteny block is shown as this black triangle, and the 2 topological domains are shown here. If we zoom into the domain that contains FOXA1, we can see that FOXA1 is in the center of the domain, and it's kind of flanked by interactions between the genes on both sides. An important thing about this locus is that, very similar to the Hox-D locus, the expression of the genes in this region is very tightly controlled. So for example, PAX9 is really expressed in only one tissue. FOXA1 is really expressed in only one tissue, prostate, well, maybe also breast, at least some cells in the breast tissue. And the genes and the tissues do not match up. If we look at which genes are co-expressed together with FOXA1, we find that the gene that is most frequently co-expressed with FOXA1, here are the FOXA1 positive tissues. The most expressed gene is TTC6, which allows us to propose at least a very early model wherein FOXA1, TTC6 are part of a regulatory domain, and importantly, MIPL1, the promoter of MIPL1, is not part of the domain, which is explained by the fact that which is supported by the fact that FOXA1 and MIPL are not co-expressed. So to understand this better, we decided we need some better technology. We went with the whole genome sequencing 10x linked read technology. I'll not explain this in detail, but what this technology allows us to do is it allows us to sequence very long, high molecular weight DNA molecules and barcode each molecule with a unique set of barcodes. And then these barcodes allow us to resolve the position of those molecules onto the genome. So here, each long molecule, which can be tens to hundreds of kb size, is shown in a different color. And this allows us to do 2 things. It allows us to phase the structural information. It allows us to detect structural information first at base level resolution, but also with much higher sensitivity compared to both whole genome sequencing and RNA-seq, and also provides us phasing information, which means it will allow us to determine which allele is rearranged, and in the case if there are multiple rearrangements, whether they affect the same or different alleles. So this is what we initially got from this analysis. Here we can see one of our model cell lines, which had a known rearrangement in the FOXA1 locus. The sequencing revealed that it's actually 2 events, which was previously unknown. There is a tandem duplication and also a translocation, which was known between chromosome 14 and the ETV1 gene, as I mentioned before. So we looked at the phasing information, and we were very surprised to see that actually both alleles are rearranged in that setting. Allele 1 has a tandem duplication, whereas allele 2 has a breakpoint, which separates FOXA1 from its enhancer. We also looked at a different cell line, which also happens to have the FOXA1 locus rearrangement. It's LINCAP. Here we detected the same translocation between chromosome 14 and chromosome 7, but we also detected a deletion on chromosome 7. When we actually looked at this closer, the deletion turned out not to be a deletion, but rather the whole event was a compound event, where a chunk of chromosome 7 was inserted in the FOXA1 locus, and a chunk of chromosome 14 from the FOXA1 locus was inserted into chromosome 7. So it's a balanced insertional translocation, which also explained, which also was associated with a small loss of a genomic region. So we then asked the question, what were the transcriptional implications of those rearrangements? And here we were quite lucky, because although FOXA1 is a very small gene, in one of the cell line, it had probably either a mutation or a very, very rare SNP. And in the other cell line, it had 2 heterozygous SNPs, which allowed us to determine whether the SNPs that are expressed or the SNPs that are not expressed are associated with the rearrangements that occurred. So for all of the SNPs in all of the genes, we found that the rearranged allele, the allele that was associated with the fusion, was not expressed. The only allele that was expressed was the allele that was either tandem duplicated or not rearranged. So here you can see that for this locus, for example, we have 2 genotypes, the C and the G, and the C genotype doesn't show almost any expression. We also went to see whether this could be confirmed in a real clinical sample. We sought the help of a company called Dovetail Genomics. This company knows how to do high-C, but can use FFPE tissues from clinical specimens. And this allowed us to see that actually the rearrangement, which is in this particular case for that sample, also a tandem duplication, that this results in a change in the topological structure of that locus. So here you can see again the FOXA1 locus. FOXA1 would be here in the middle. Interactions, as predicted by the high-C data from the cell lines, would span this locus, whereas we only observed these interactions, which allows us to propose a model wherein we have the FOXA1 locus, and at least in the cell lines and in the tissues that we have studied so far, one of the alleles is rearranged, and the hypothetic enhancer in that locus becomes the driver for the ETV1 gene, whereas the other allele loses access to its enhancer and also loses expression. The other, the FOXA1 gene loses access to its enhancer and its expression. On the other hand, the other allele is subject to additional rearrangements. That's the case of the second allele to restore the expression of FOXA1. Because one important thing about FOXA1 is that it seems to be a very a gene with whose dosage is very controlled. So it's first a haploinsufficient gene, but also overexpression of FOXA1 leads to drastic phenotypes in the case of prostate cancer cell death. So were other types of events like that reported? Not quite. So if we look at recurrent positional effects in cancer, this is the whole list I could amass. And most of them are related to so-called enhancer swaps, where the structural variant causes the expression of a gene by the acquisition of a foreign enhancer. The only thing that comes really close to FOXA1 rearrangements are rearrangements in the MYC locus, and that also only in the multiple myelomas, where it's also like for FOXA1 a late event. So I guess it remains to be determined whether this is a complete list. I think that this is the very, very tip of the iceberg, because previously we were not really looking for them. But also it remains to be determined how frequent those complex rearrangements, like in the FOXA1 locus, are. So to just conclude my talk, I think position effects provide a framework for the interpretation of structural variation. We can integrate and think about structural variations by integrating genetic recurrence, which we can do from whole genome sequencing data, whole exome sequencing data, and RNA. We can integrate on top of it the transcriptional changes, the topological domains, I think very importantly evolutionary relationships, but also the developmental and clinical genetics. So insights into how to deal with the germline congenital diseases really can inform how we think about structural rearrangements in cancer. The other thing I would like to emphasize that epistasis or synthetic dependencies between SNVs and drivers and CNVs are underexplored. So really nobody showed conclusively how p53 loss leads to genomic instability. Those types of association might provide us insight both on the oncogenic role of the mutations, but also on the oncogenic roles of the CNVs. So for example, the recurrent loss on 6p, it might tell us something actually about what the oncogenic role of ERK is, which has eluded explanation for the last 10 years. And also, we may through this approach identify new molecular subtypes of cancer. Perhaps prostate cancer with a 6p loss should be considered a molecular subtype, and perhaps it is associated with differences in prognosis, and we are currently doing those types of analysis. I would like to emphasize that at least for cancer, link-3 technology is a game changer. Haplotype-resolved SVs are really useful, and also, at least as computational tools mature, these will provide us with improved sensitivity of structural variant detection. I would like to emphasize one idea that was very, very difficult to address without linked sequencing. This is mitotic crossing over in cancer, right? We often talk about non-allelic homologous recombination in cancer, but what about allelic homologous recombination in cancer? These changes cannot be detected without phasing information provided by technologies like this. And I would also like to mention that we are currently a little bit limited. We have no good statistical framework to quantify recurrence at the structural level, right? Probably it shouldn't be arbitrary windows in the genomes. It should be something more like a topological domain, or maybe not, maybe a replication domain. And also our current approaches, or at least the ones that were published to detect rearrangements from whole genome sequencing data, are limited. And which was a very learning experience for me. I was not really aware that our understanding of germline structural variation in humans is so poor. So with that, I would like to first I would like to thank the students I'm working with. They make everything fun, so I would like to thank them. And also the small bioinformatics staff who works with me. They allow me to focus on many things, and they are really a big help. So with that, I would like to thank all the other people. I would like to thank Arul, the genomics group, the bioinformatics group, and of course the clinicians, pathologists, biologists, and of course my collaborators here at the bioinformatics department, and also the funding. Thank you.
So in the CDK12 mutant tumors, you said with RNA-seq, you
Yeah.
found a specific pattern of gene expression that was different.
Yeah.
Does that have anything to do with gene size? Did
Yeah,
small
so-
size?
Yeah, so we tested for this. So the literature suggests that these should be long genes that are differentially expressed because CDK12 has been implicated in the splicing of the last exon, particularly or splicing of the last intron. We didn't find the signal. I'm not totally confident that the signal isn't there because we are still working with clinical samples and we cannot generate the knockout cell lines because they are not viable. So we cannot really rule this out completely, but there was no statistical difference. The genes as a whole, they didn't fall into any kind of gene set that might be enriched. So there's nothing I could tell you about those genes. They, as far as I can tell, are pretty random. No, in terms of size, they are average.
You know, do you think that, you know, it looked like you have enhanced Somatic, it's going to be a big deal, you know, and that, yeah, I think that's the tip of the iceberg thing you said is probably true. And it looks like you might be able to discover a new class of regulatory variants the way that Alan Boyle and others in the department here have done so, and our group and Steve and others. Looks like a nice, you know, and it opens the door for work with the topologically associated domains and all the TADs, and I think that's probably going to be a meaningful structural component in your analysis going forward, including the high-C measurements. So when you send things to Dovetail, how does that work? I mean, is that, hadn't heard about that.
Well, we are still in the very early process. We still are working out, you know, how to do this. The Dovetail has their emphasis on detecting structural variants. They think that's their core strength. I think their core strength is actually doing high-C and FFP clinical specimens, so it's a challenging collaboration from that perspective. But I think definitely they know their molecular biology, which is even for advanced labs often very difficult to achieve. So they definitely know what they're doing.
Yeah, it looks like a good direction.
Yeah.
Where do you see all this going in that? You know, just in terms of your career development and how this plays into your the framework of your thinking. Where do you find the next? I thought the conclusions were solid and expansive, but where do you want to take this, Marcin?
Well, I think right now we are just beginning to be allowed to use the ICGC structural whole genome sequencing data sets. And from everything that I've seen published, preprinted, the approaches that have been used didn't discover anything. So I'll
right.
So that I think we have pretty strong evidence for the rearrangements in the FOXA1 locus. And prostate cancer was already sequenced quite a number of times using whole genome sequencing data. So those rearrangements are there. They were just missed. They are missed over and over and over again. People sequence more, but do not detect them. And I think this is mostly a result of a lack of framework for their aggregation. The individual events are detected, the recurrence is not. So I think one of the biggest challenges moving forward will actually not be the improvements in detection, because this is really limited by the technology that we are using, but really the algorithms to determine recurrence, what is recurrent, what isn't recurrent, and how we can use other types of information to actually inform those algorithms. A recurrence analysis algorithm shouldn't take just BAM files or aligns or cold structural variant. It needs to take into account the biology. It needs to take into account the expression. And once it does it, I think that our sensitivity will improve.
Right, so modeling in the biological context to inform the analysis.
Well,
and I guess the one thing that could be, because you mentioned you were going to speak about the algorithms, and you know, I guess I'm coming from more of a computer science background, but I did see the one diagram where it could be construed as an algorithm, so that would be a good thing to continue to to discuss a little bit.
Yeah, so
Doesn't have to be now, but
yeah,
it'd be
so
good.
I guess the sequence analysis is all the same. So all the breakpoint assembly algorithms that go into RNA-seq could be applied to whole genome sequencing data or linked read data for that matter. So these things are pretty similar, but I think it's the added information that is valuable. So for example, if we interpret the variants in a given cancer type, we should probably anchor them on at least the topological domains on this cancer type as the unit of recurrence.
Yeah.
A unit of recurrence.
That's a big implication, I think, for this work. And it's definitely there, but that as a future direction, that's can certainly be fleshed out.
Yeah.
And this, in the context of all the rearrangements, that is going to be a complex and interesting problem.
I think it's also, I mean, ultimately it's not detecting rearrangements for the sake of rearrangements. Ultimately, in this small deleted 6p locus, this narrows down the number of genes to 20. We don't know yet which 20, but it becomes very feasible to actually functionally implicate a specific gene.
Well, you mentioned the gene set work, but I think, some recent studies that we've been doing has been suggesting that those gene sets would live within a topologically associated domain. And some of the genes, if you get the topology down, that might help you understand where the fusion elements are placed and where the genes are. And it could just simply be a part of, if you think back to the kind of standard diagrams of DNA and then nucleosomes and chromatin and then heterochromatin and loops, it's pretty clear that that diagram isn't quite right. We can maybe talk about it at dinner, but I believe the topologically associated domains are going to be fitting in there. Enhancer promoter pairs, you have those kind of loops, how those are organized into TADs, and then how those create, either interphase chromosome territories or banded metaphase chromosomes. I think if we redraw those, we could reinterpret a lot of the functions that you're seeing in the cancer context or in the normal context in a more sophisticated way. And I think your work is pointing in that direction. It's very good. Trying my best. Okay, anything else? Well, let's please thank Marcin again. That was really a tour de force of bioinformatics analysis, and we really appreciate it. We all learned something from you. Thank you very much. Yeah. I thought it would be fun to take you to Amadeus, but maybe you go there every weekday.
No, I never went.